That is the jam. That's the jam of just my life. I'll literally play that song in the car and just be belting out Bless the Rains down in Africa. Okay, so on to biology. So we're continuing from last time on. We discussed all the cell organelles in animal cells and plant cells. They're all the same, except that plant cells have those additional structures, cell wall, chloroplast organelle, where photosynthesis goes down, and the vacuole that stores excess water. And we're going to continue today by talking about, so we mentioned animal cells and plant cells. Now, both of these type cells of animals and plants belong to a category or a type. The cell type is eukaryotic. And we're going to talk about the difference between eukaryotic and prokaryotic cells. And they basically, these two categories of cell type define all the cells that we find on Earth. And they're so starkly different, just so drastically different, that they put them in two separate categories. But you won't find an organism on this planet that has a cell type other than prokaryotic or eukaryotic. So we'll take a look at that today to start off. And then we're going to go into energy and enzymes, which I forget how many lectures I have devoted to that, but we'll see how far we get and should be working. I think we have 2, so that's really good. All right, so let's get started here. So like I said, 2 different kinds of cells in any type of organism. Now, keep in mind, no organism has both prokaryotic and eukaryotic cells. So animals are eukaryotes, we sometimes call. So all animals, all the cells are of eukaryotic type. There you go. And then we'll find that there are these organisms that have prokaryotic cells, but as we'll see when we draw on the board, that all prokaryotes, all organisms made of prokaryotic cells, are actually unicellular. So I say prokaryotic cells, but there's no organism made of more than one prokaryotic cell. So bacteria and these organisms called archaea, they're prokaryotes, but they're all single-celled. So like Salmonella bacteria, E. coli, Vibrio cholerae, the bacterium that causes cholera, tuberculosis, all these different bacterial species are single-celled. So if you have a colony of bacteria, it's not one organism, it's millions of bacterial cells in this colony, each one being an individual organism, a single cell. So that's what we're gonna look at. And we'll also find that there are single-celled eukaryotes as well as, of course, like us, multicellular, because we're made of trillions of cells, right? So let's go ahead and do a comparison and define what it means to be prokaryotic organism or eukaryotic. Okay. And also, just a quick announcement, mention or whatever. Just with the lab, I can answer questions after the lecture today. Just like usual, I kind of hang out for any questions on the lab due tomorrow. So, okay, I'm going to write prokaryotes. So it's kind of the word we use for prokaryotic organisms. We call them prokaryotes and eukaryotes. And we're going to define what is different in the cells that we see, what kind of structures, what kind of what kind of structures we see that are different between prokaryotic cells and eukaryotic cells. Okay. Let's get this out of the way. Okay, so the first thing I want to mention, or we'll start with prokaryotes. Might as well. It's on the left here. Actually, before I dissect this word, let me put some examples of prokaryotic organisms. Actually, the only two domains of life that are prokaryotic, as I mentioned, are bacteria and organisms called archaea. And all you have to know is that these are domains of organisms, two domains here. So bacteria encompasses thousands of bacterial species, archaea encompasses thousands of archaean species. They're both single-celled microorganisms. I'm going to go shut my window because my neighbors are loud. Okay. So all bacteria, all archaean species are single-celled microorganisms. If you were alive during the 70s, I wasn't. There was actually a headline when in the New York Times, just like front page news worthy. I mean, science and biology, we rarely make front page news. And when archaea were discovered, they actually thought archaea were bacteria. They were prokaryotic, but they're actually different enough. These scientists discovered them, I believe, in hot springs in Yellowstone. They were different enough to become their own domain of life. Different enough from bacteria. So it was like a huge, huge deal back then when that discovery was made. Okay. And as you saw in my slide, prokaryotic cells, they're much smaller. Oh, there's a picture of one, but here's a better representation. Prokaryotic cells compared to eukaryotic, they're smaller, right? Very small. They're simpler, not as complex, don't have as much going on. And we're going to kind of define what is present in prokaryotic cells and what isn't. And I'll start by dissecting the word prokaryotic or prokaryote. So the prefix pro here, kind of like a prologue of a book, means before, right? It's before the book. So there'll be a prologue. My voice cracked for the first time in a long time. Anyways, ignore that. Like prologue, before. And so karyo actually refers to nucleus, which we discussed last time, is that organelle that houses the genetic material, DNA. And pro is before, right? So this means before nucleus. And this is a very good description of a prokaryote, because not only do they lack a nucleus, so we're going to write cells with no nucleus, but prokaryotes actually existed on the planet before eukaryotes. And we'll find that eukaryotes, of course, have a nucleus. But they actually existed before a nucleus was even a thing. It wasn't an actual membrane enclosed organelle. And because of that, all life has DNA, unless you think viruses are alive, some of which don't have DNA. But all life officially has DNA as genetic material. And so the DNA of prokaryotes is actually just floating around in the cytoplasm. So we'll just write DNA located in cytoplasm. So it's not as protected. And like I said, these cells are smaller, they're simpler. So the DNA, all of the genetic material, we call the genome of an organism. For reference, our genome is huge. It's about 3 billion nucleotides long, massive. But certain bacteria and archaea, their genomes are like 1,000, if that. So much smaller. They usually have, yeah, so anyways, just floating there. The second thing that's characteristic of prokaryotes, something else they don't have, as we talked about last time, we discussed all these organelles, right, with specific functions. Prokaryotes don't have most of them. And any of them that have membranes enclosing them, they do not have. So I'm going to write lack membrane-bound organelles. So last time I mentioned that there's an organelle that isn't membrane-bound. And we're going to come back to that. We're going to come back to that because that organelle you will find in prokaryotes. It's essential to all life. All cells on the planet have a certain organelle, but it's not membrane-bound. So I'll underline that. The prokaryotes do not have any membrane-bound organelles. So they don't have lysosomes, mitochondria, Golgi, smooth, rough ER, chloroplasts. They don't have any of those. They basically only have DNA floating around and one other thing that I'll mention shortly. Okay. I wonder if I can fit eukaryotes here. I'll try. Scoot it up. I kind of went over too much, but let's talk about eukaryotes. So if all life on the planet is either prokaryotic or eukaryotic, basically we're going to write everything else here that exists in our world. That would be us, animals, right? We are animals. That would be plants. Anyone know what else exists on our planet besides animals and plants? Bacteria and archaea? Fungi, absolutely. Fungi like yeast and mushrooms and molds. Okay. And then there's actually one more group or kingdom of eukaryotes that you might not have heard of. They're lesser known, but they include amoebas, they include algae, because algae wouldn't be considered a plant. It's kind of the misfit group, and they're called protists. So protists are eukaryotic, and they're actually an ancient kingdom. They're the first eukaryotes that existed on the planet. I think the first one was around 1.5 billion years ago. So they predated all three other kingdoms. And they include, again, amoebas, plankton, phytoplankton in the ocean, seaweed. those are protists. And so protists are kind of like the misfit group. They're organisms that aren't really true animals, aren't true plants or true fungi. They kind of resemble them though, but they're unique in such a way that they couldn't be put into those groups. Okay. So basically we're gonna write the opposite here. First criterion of eukaryotes, cells have a nucleus. So if we actually break down the word eukaryote, this U prefix means true. And then karyo again refers to nucleus. True nucleus, there is a nucleus that's going to house the DNA. That houses DNA. And then the other criterion, unlike these guys, they do have membrane-bound organelles. All of the ones we mentioned last time have membrane-bound organelles. So our eukaryotic cells have lysosomes, they have endoplasmic reticulum, mitochondria, etc. Okay. Now, I'm going to use a different color that I haven't used yet to emphasize this last point. Let's go with blue. It's one organelle I haven't said, ribosomes. Ribosomes, remember, were the organelle responsible for where proteins are made. Proteins are so vital to life, no matter what kind of organism you are. You need proteins. And so ribosomes are not membrane bound. So I'm very specific when I say that the prokaryotes don't have membrane-bound organelles, but they do have ribosomes. And so do eukaryotes. So I'm going to write under both columns here, have ribosomes. Ribosomes are not membrane-bound. They're actually made of a type of nucleic acid called RNA. And they're not surrounded by a membrane. Remember, our rough ER was coded in ribosomes? Well, the prokaryotes don't have rough ER, but they have ribosomes floating around in their cytoplasm. All right. That's it for cells. We're moving on. So any questions before I move on to energy? Energy and enzymes. The DNA. We are starting a new lecture. Yeah, this is kind of finishing up last time because we ran out of time. So we're about to start the next set of PowerPoint slides on energy and enzymes. Where's the DNA floating? So the DNA is floating in, so I'll actually show you the picture. There it is. So here's our prokaryotic cell. And you can see the DNA is just floating around here in the cytoplasm, in the fluid, right? Just suspended there in the jelly-like fluid substance within the prokaryotic cell. But then, of course, our eukaryotic cells, as shown here, they have all these complex membrane-bound organelles, and then they have this massive nucleus, and that's where you'll find the DNA. Oh, there you go. That's it. Okay. Well, I think I got it up, the other presentation. Cool. Energy and enzymes. All right. So this is, we're about to enter a territory that's pretty different than what we've been talking about, in the sense that we're going to be talking about some physics concepts, again, surface level, not too crazy. And then we're going to be discussing, throughout this unit, actually, we're going to be discussing some pretty intense but intricate and amazing biochemistry. And it's very different than not just the previous units, but future units too. It's unique in that way. But we're going to start today's lecture by doing some physics and talking about this principle of thermodynamics. Of course, this is biology, and you know me, I'm always going to find, ways to relate it. And part of why I love biology is this fact that it's probably the only science where you can talk about physics and chemistry as we've done, because it's all so interrelated and so relevant. Whereas if you're just talking physics, it's hard to integrate biology sometimes, or chemistry into biology. So we're going to find that within our cells, these physics concepts are extremely important. Well, just one in particular, because physics is not my forte, but the thing that I find most fascinating is this concept in thermodynamics. Okay, so let's begin. Just reminding myself here. Okay, so if we start by looking at the slide, you'll see some energy transformations. This is going to touch upon this thermodynamic principle that we're going to talk about, but I just want you to take a look at what's happening here. There's energy from the sun, light energy, that plants use to photosynthesize. They actually transform this energy to a different form, which is actually sugars stored in the plant. So again, the plants are using photosynthesis, this process within their cells, to convert light energy to chemical energy stored within sugar molecules. And then, because ultimately everything, all food, including if you eat meat, comes from this, and I won't go into detail, but from this process, essentially, as energy from the sun is stored as sugars, and like in this ice cream cone, it can then be converted to a different form of energy called kinetic energy. And that's what we do a lot. So we eat food, right? The reason being is because, and this unit is really devoted to this process of extracting and using the energy, chemical energy stored in food molecules, like sugar, like fat, even protein. And converting it to kinetic energy. See, as I move my arms around, I'm contracting my muscles, And that's an energy requiring process. And that energy ultimately comes from the food I ate. And I have to extract that energy from that food. And what I'm doing is converting that chemical energy in the food molecules that I eat to this energy of motion, kinetic energy, sort of releasing it from the molecules to move my arms, using it. And so the point here is that we're doing energy conversions. We're transforming energy to different forms, different types, right? That's the main point here. So we're going to start by saying, let's define energy as biologists do, as this ability to do work. And when I say do work, in science, specifically what work means is to move objects or move matter, move anything against an opposing force, like gravity, like friction. It's basically an doing an energy requiring process. That's work. So it's a little trippy to think about doing an energy requiring process when it's the ability to do an energy requiring process. But work is this idea of if you do work, then you are doing an energy requiring process, something that requires energy input because you're going against a force. So you need some energy input to do that. It doesn't happen spontaneously, as we say, or just without any energy. Okay. So to do work, for me to go on a run and overcome the friction, to overcome gravity and go up a hill, I need energy, of course, and energy will give me that ability, right? So what I'm doing is I'm doing an energy conversion, a transformation. I'm transforming what we call potential energy, which you could think of as stored energy, sometimes called potential chemical energy. So you could even add chemical energy here, as we saw with the ice cream. There's stored energy in those molecules, specifically in the bonds between the atoms in sugar molecules, for example. So I'm going to also write example is food. Don't really need. So food we talked about disaccharides, starch, triglycerides in butter and cheese has chemical potential energy in it. Potential energy stored in it. And what we are doing all the time is converting it, releasing it, exploiting it into kinetic energy, the energy of motion. This is the energy of motion. Now, I cannot just move my arms and create energy out of nowhere. I got to get it from somewhere else. Like, that's the key. I can't just create energy out of nothing. You also cannot destroy energy and get rid of it. It has to transform to a different form. Energy cannot be created and it can't be destroyed in this universe. It will exist in this realm in some way. And we call this the first law of thermodynamics. This is the first law. I think I'm running out of room here. So I'm going to erase this and rewrite. So the first law of thermodynamics states energy cannot be created or destroyed. It can only be converted from one form to another or transformed. It can only be transformed from one form to another or type of energy. So these energy, this question. So an example of potential energy transforming to kinetic energy. It's a great question. The best example, most relevant to this unit, is when we eat ice cream and the sugars are entering our body. Those sugar molecules have chemical potential energy in them. And what we will do is this process of breaking the molecules apart further, picking them apart, breaking them down, and releasing that energy. That released energy can be used to move your arms, to do anything that an energy requiring process in the cell. So movement, active transport that we've talked about, right? Yeah, we did. That's energy requiring. Changing the shape of certain proteins, sort of kinetic energy. So really taking that potential stored energy in food molecules and releasing it to and moving the molecules of movement, giving them more movement, which allows them to oppose forces like friction and gravity. That's probably the best example I can think of. And it's the most relevant. It's the one we're going to explore in detail. Another thing we use energy for is building, as we learned with dehydration synthesis. Well, that process requires energy, building those molecules. Did we talk about that yet? I forget. I don't know if I mentioned the energy part of it. No, I haven't. We're going to talk about that next, actually. So it's a good segue for the next slide. Metabolism. That's what I'm hinting at. When we break down food, when we build molecules, it's part of these metabolic pathways in our cells, in this entire concept of metabolism. Let's define metabolism. Metabolism, chemical reactions. That occur inside the cell, all of our cells, to either use or produce energy. And more specifically, when I say energy, who remembers the energy currency of the cell? What molecule is that energy-rich molecule that we've looked at? And I believe we're going to draw it, yeah, on the next slide. I think I mentioned it at the end of the biological molecules lecture. It was a nucleic acid. ATP, absolutely. So we're going to be looking at pathways and chemical reactions. Pathway meaning a series of chemical reactions, one after the other, that are going to use energy in the form of ATP or produce ATP molecules. And so when I said we're going to exploit the energy in sugar molecules in food, we're actually going to produce ATP and ultimately from our food molecules. And that ATP can be used for processes that require energy, like movement, muscle contraction. Okay, first thing, few terms we're gonna look at. So we'll go back to the slide here. Two types of metabolic pathways. We have anabolic. So an anabolic pathway is when small molecules are built into larger ones. So relevant to what we've talked about, we would be, combining monomers to build polymers. And energy is required. So I'm actually going to rewrite anabolic here. And I'm going to write monomers. Should have wrote combined monomer. Well, yeah, let's do that. Combined monomers which will create a polymer or polymers. So an example of this, just use the monomer for proteins. What was the monomer for protein? What was our monomer unit? Yeah, amino acids. So amino acids, when we link them together to build a protein. Now, it does say, right, that energy is required for this process, and we call that endergonic. So anabolic reactions are endergonic. Who's knocking on my door? Whatever. Endergonic, meaning energy is required or energy input required. So anabolic reactions, when we are building larger molecules, polymers, with these component monomers, putting them together, that's an energy requiring process, and we call that endergonic. the flip side, catabolic. So the converse of this would be a catabolic reaction. Which is just the reverse. We're going to break down larger molecules into smaller ones. And instead of energy input, energy is released from a catabolic reaction. So let's write, oh, I totally ran out of room. I have an idea. I'm just gonna place this. Catabolic. I'm gonna write break down polymers into monomers. So in a catabolic process, we're breaking apart a larger polymer molecule into individual monomers. And the best example, as I mentioned, would be this process of breaking down sugar molecules from like an ice cream into monosaccharides, for instance, digestion. So let's do starch, which was a large polysaccharide molecule, into monosaccharides, like glucose. And we're going to talk a lot about glucose in this unit. So I'll even add like example glucose. And what do you think it's called when energy is released? Not endergonic, but energy is not entering, it's exiting. Yeah, it's exergonic. Catabolic reactions are exergonic, meaning they're going to release energy. And so, like I said, this digestion of food, when we break it down, we can use that potential chemical energy in the food molecules, and the energy that's released, we're going to be, it's going to be, I would say, in the form of ATP. We're going to be creating ATP molecules from, and this whole, in a sort of factory-like process of generating ATP from food. It's the heart of what we call cellular respiration, which is a large part of this lecture series. Definition for question mark catabolic. Catabolic, large molecules broken down into small ones. Energy is released, but really when we think of catabolic, we are breaking down a larger molecule into smaller ones, breaking apart a polymer into monomers, and there's going to be an energy release as well. Is it similar to endothermic and exothermic? Yes, it is. Yeah, I mean, I'd have to re-look at the actual chemistry of that, but I think it is, but I think exothermic and endothermic are a little more specific, and they're talking about heat energy. And so there's always going to be some heat released from these reactions or heat input. But yeah, so the concept is similar, but I would just, yeah, not think about heat for this class. At least not yet. Yeah, I think that's more with chemistry, when like a lot of it's released as heat or a lot of heat input is required. But I'll have to look into that again. Okay, ATP. Let's take a look. It's similar vaguely. Okay. Yeah, so I wouldn't even think of it as that. Okay, ATP. That's ATP. That's what ATP looks like. And we're going to kind of talk about a characteristic or two of ATP molecules. So ATP stands for adenosine triphosphate. And I'll get to the adenosine part shortly, but the tri, here's the ATP, triphosphate refers to tri, like tricycle, 3 wheels. Triphosphate means 3 phosphates, 3 phosphate groups. And so I want to connect and show you how and why an ATP molecule has a lot of energy. Why it's considered a high energy molecule. And so for simplicity, the adenosine part of the molecule, I'm just going to have this A here. And as I've said, 3 phosphates are attached. So 1, 2, 3. And when we looked at our cell membrane, remember it was a phospholipid, and that phosphate group had a very negative charge associated with it, right? So do these. These phosphate groups have negative charges. So I'm going to write a little negative on each of these guys. Now, this molecule of ATP has a lot of chemical potential energy inside of it, specifically between the bonds. Now, the reason is, as we learned with covalent bonds, we're sharing electrons. And so these phosphate groups are close together, of course, they're bonded together. And there are negative charges associated with these phosphates. And we know that opposites attract. Opposites want to be next to each other, plus and minus. That's a more stable situation. That the universe likes that. But when we have all these negative groups close together like this, they don't like it. Like charges repel. There's this repulsion. And so this guy is kind of barely hanging on. He's barely sticking together, and there's just this force, this tension between the bonds. And actually, this bond in particular is the highest energy bond. So it's a high energy bond here in this third phosphate bond here. It makes this molecule very unstable, high energy, unstable. These electron repulsion, they're dying to come apart. And I'm not sure why this is. You don't have to know why, but it's just a fact that when you have molecules with electrons so close to each other and repelling each other, there's a lot of energy there. It creates this high energy state, and this ATP has a high energy state. So let's break it apart. Let's break off that terminal phosphate group, that third phosphate. So we have the A adenosine portion, and now we only have 2 phosphate groups. And I broke one off. I kind of ran out of room here, but I'll just continue down here. A PI. So this stands for inorganic phosphate. All that means is it's not connected to anything. It's not connected to an organic molecule, carbon atoms. So this is just a free phosphate group that we broke off. And here's the concept. Here's the relevance of that first law of thermodynamics. Energy cannot be created from nothing, and it can't be destroyed. So this energy in this high energy molecule, when I broke it off, and this is much more stable. This ADP, we call it, adenosine diphosphate with 2 phosphates. Much more stable, lower energy, and that energy is released. It's not destroyed. It's not created from nothing, but it comes out. So we get energy out of this process. So I'm going to write plus energy. So whenever you're going from a higher energy state to a lower one, that energy just doesn't disappear, it's released. And this is also a catabolic process. We're breaking apart a smaller molecule into 2 smaller ones, the phosphate and the ADP. And so there's this release of energy. It's exergonic. And our cells can use that energy. So you see this reaction occur a lot in energy processes in the cell. And then, of course, and we'll see this too, we do this too, we want to build ATP molecules because we want to use them eventually. And so we'll actually do the reverse reaction sometimes. So this way. And we'll take an ADP and a phosphate. But of course, that requires energy, right? If you want to create a high energy molecule, you're going to have to put energy into it. And if you think of it like this, you're trying to stick on this negatively charged phosphate to this ADP, which already has 2 negatively charged molecules. You're going to need some energy input. It doesn't want to do that. So if you have this negatively charged ADP and you're like, oh, I just want to put 1 more negative on there, there's repulsion. You're going to need to force it on there. You're going to need an energy input to create ATP again. And so creating ATP requires energy. And we'll see that that energy comes from our food. Okay, how are we doing on time? Okay, we're good. Okay, so let's just take a look at a detailed version of ATP. So I just wanted to show you kind of what it looks like, because it looks like a nucleotide. It is a nucleotide. It just has two additional phosphates bonded to this one. And so it's an ATP molecule, and the adenosine part is only referring to this adenine nitrogenous base and this ribose sugar together. So that's the adenosine just means these two together. That's adenosine. And there's your triphosphate. So I kind of alluded earlier to what is required in the cell, what processes, mechanisms require ATP in our cells. Like, why do we need ATP? Why does all life on the planet need ATP? And all life does need ATP. There's a few reasons. What do cells use ATP for? First, let me ask, are there any questions before I talk about that? Okay. Let's look at a few reasons why cells use adenosine triphosphate. And you can see that there's all these ATPs, right, required, except this picture I pulled, but I'll explain it. Let's go ahead and, yeah, actually all the plant cells that do photosynthesis, it's a great question. Photosynthesis, while it uses light energy, there's also ATP involved, because there's an anabolic component of building sugars from smaller molecules. So there's going to be some ATP input. So yeah, that's a good question. Okay. So let's list some reasons why cells use ATP. I talked a little bit about movement. So you can see that in the second picture. We've talked a bit about active transport, right? Going from low to high, that transport mechanism. So that requires ATP. So let's write active transport. Let's write muscle contraction. Let's write cell movement. So I'll move out of the way. So that's actually shown on the slide. So here's our active transport, right? Sodium potassium pump requires ATP. Ion pump, whatever. ATP is required for active transport. This is actually, so our muscles are made of these two fiber proteins called myosin and actin. So to slide them and contract the muscle requires ATP molecules. It's a going against friction type thing. Did my share go away? Okay. Cell movement, hugely important for some of our cells. Sperm cells for males actually, to move, the tail needs ATP to, I forget the exact, they're motor proteins that are moving around and they require ATP. It's going against friction, and of course that requires energy. And then this cell is called a white blood cell, and it requires ATP. So in defending our body against constantly exposed viruses and bacteria in the air that we breathe in, or we get a cut, we have constantly patrolling white blood cells looking for them. Moving around constantly and entering and exiting tissues. And the way they do this is by breaking down and squeezing and expanding and contracting, all this cool stuff using ATP, cell migration. So cell movement is going to require ATP. And then this is a cool, complicated thing, but I circled this bottom part here because, well, there's two more things. Sorry, I'm going out of order here, but we mentioned anabolic processes, right? Process where we're going to be building macromolecules, and there's a lot of them that we do. A lot of molecules need to be built. And then the last one would be activation of enzymes. Which you've seen a bit about enzymes now in the lab, but like this lecture says, it's energy and enzymes. So we're going to be talking about enzymes very soon. Okay, and just pictured here, some important anabolic processes, synthesizing DNA, synthesizing glycogen for storage of sugars in our liver and our muscle, synthesizing fat, and proteins, as we saw as our example. And yeah, all of these anabolic pathways, as we saw, require energy input. And this picture was the activation of an enzyme. Enzymes, perfect segue. So let's start talking about our enzymes here. 405, okay. Okay, so what are enzymes? What are they? We've looked at the lab now, but I've mentioned it's out of order for some reason. The lab should be next week. Should have done cells and microscope. I don't know. But basically oh, here you go. They're proteins. Enzymes are proteins, and they catalyze chemical reactions. They cause them to happen. They're necessary for all of these. So every single biochemical reaction, breaking down sugar, building fats, building proteins, enzymes are required for all of these. Okay. Back to the slide. Okay. So we're going to be looking at what we call a substrate and products. And essentially, there's this, for every chemical reaction, there's what we call an activation energy, whether it's a small or a larger one. Enzymes, the official role is overcoming that activation energy barrier by lowering it. So there's this activation required, this tiny or large, if it's an endergonic energy requiring process, like an anabolic one, you might need additional energy, but enzymes are going to lower it no matter what, a little bit, or yeah, a little bit, enough. But like I said, sometimes you need additional energy input as ATP to do something that's endergonic. Okay. So here's an example that I like to use. I believe we've talked about it before when we talked about carbohydrates, but the digestion of lactose. So lactose was a disaccharide, right? So it was a disaccharide, two sugars covalently bound together. And then we have a reaction arrow and we're breaking it down into glucose and the other monosaccharide called galactose. And this chemical reaction requires an enzyme. And we write the enzyme on top of the reaction arrow. And for this one specifically, the enzyme is called lactase. So this is our enzyme. It's a protein. And today we're gonna, well, how long do we have? We're gonna begin to talk about the last 5 minutes. We're gonna begin to talk about how lactase is going to catalyze this reaction and how lactase is going to play a role in converting lactose into the products glucose and galactose. Whenever we're talking about an enzyme, the reactant, the starting material, whenever we're dealing with enzymes, we use a different word called a substrate for the enzyme. It's used because substrate is just a word in science that's sometimes used for when another thing is attaching to it. So the enzyme we'll see will attach or bind to the substrate molecule, or if you have multiple reactants, multiple substrates. And then always the right side, the ending molecules, whatever you get, are called products or a single product. Here we have two, so products. I'm actually thinking here might be a good place to stop. Let's see, it's no, we have 5 minutes. We'll keep going. I think I can start to talk about the next concept. Actually, I have a great idea. The next slide says naming enzymes, right? So let's talk about that. And then we'll do enzyme function and how enzymes, what they're doing, we'll do next time. But let's just talk about how we name them. So let me just write naming enzymes. So you'll notice this enzyme here, lactase, ends with ASE, right? ACE. So 99% of the time, the ending is going to end in ASE. So I'll do ASE. And then like a few proteins, enzymes are called or end in IN. Such as like pepsin. Pepsin is a digestive enzyme. So is trypsin. I'm not sure why, but just a few that end in IN. 99%, however, end in ASE. So there are so many important enzymes. So I'll just name a few. So examples, ATP synthase, lactase, of course, we looked at. What's another good? Protease breaks down proteins. dehydrogenases, so dehydrogenase, like alcohol dehydrogenase, etc. Is there a question? Oh, old naming conventions. Oh, interesting. Cool. Oh, lipase, like pancreatic lipase breaks down fat molecules. It's created from the pancreas. One of the major roles of the pancreas is to create and release digestive enzymes like that. So they all end in ASE. And in fact, to end today, just end a minute or two early, I'll show you that. Does anyone recognize, we looked at protein structure, that tertiary structure. So you can see the tertiary structure, but does anyone recognize that the substrate that this enzyme is binding to in the enzyme here? See how they're attached and it's like weaving through there? Does anyone recognize it? Yeah, it is. It's DNA. You bet. And so that protein is called DNA polymerase, another crucial enzyme that actually catalyzes the synthesis of DNA. So if you want to add DNA polymerase, that's another great example. So of course, as you can see, enzymes are crucial for life. They're crucial for why we can get energy from food, why we can build important molecules, detoxification of alcohol, alcohol dehydrogenase. We'll see there are other dehydrogenases in metabolism, but yeah. Okay, I think that's all I want to talk about today. What time is it? Yeah, 4:15. So any questions on the lecture or lab? I'll just pull up my chair here if you have any questions.